A Punnet Square is a great tool for predicting the outcome of a genetic cross. The key to correctly setting up a Punnet Square is knowing how the alleles are distributed during the process of meiosis. Let's take a look at how meiosis and Punnet Squares are connected. But how are those two things related? Let me introduce you to Mama Goose and Papa Goose, which are expecting. What will their goslings look like, Mom or Dad? Let's figure this out scientifically. In geese, white feathers, the big W, are dominant to brown feathers, the little w, and red beaks, the big R, are dominant to yellow beaks, the little r. Our mama goose is heterozygous for white feathers and homozygous recessive for a yellow beak. Papa goose is homozygous for brown feathers and heterozygous for a red beak. Isn't that romantic? For mama goose, meiosis will result in her beak and feather alleles being distributed in one of two ways. The gametes she produces will either have a big W, little r, or a little w, little r. Papa goose can also produce two types of gametes, little w, big R, and little w, little r. What sort of goslings will this pair produce? This egg can combine with this sperm, or that sperm, and this egg can combine with this sperm, or that sperm. This gets really confusing really fast. This is why we use Punnet Squares. The top and the side of the Punnet Square are filled out with the genotypes of the gametes that each parent can produce. Remember that because of independent assortment, each gamete gets one copy of each allele. You would not write little w, and big R, little r. Organized in this way, we can quickly and easily figure out the potential offspring from crossing these two parents. Should we tell them or keep them in suspense? Let's tell them. We know that because of independent assortment during meiosis, the gametes from these parents could combine to give us goslings that have white feathers and red beaks, white feathers and yellow beaks, brown feathers and red beaks, or brown feathers and yellow beaks.